So I will first start talking about proteomics and why is it important, and I'm going to talk about some issues with proteomic methods. Then after, I'm going to talk about the hybrid method I developed for sample preparation to get an increased number of identification. And then I'm going to talk a bit about mass spectrometry analysis, how is it done and how the quantification is done. And I'm going to show you some cell applications after by using the hybrid method just for validation purposes of the protocol. So proteomics is important, and we all know that genome contains all the genes required to build a functional organism. However, it's the only source of an information, and in order to function, it must be expressed. And this transcription of the gene starts with the gene expression, and it's followed by the translation of mRNAs, and it will be followed by the synthesis of proteins. And proteome means the complete set of proteins produced by genome at any one time. Proteins are much more complex structures because each protein can be chemically modified after the synthesis, like acetylation, phosphorylation, and so on. And they are very dynamic in cell in the sense of from the same genome, different proteins will be expressed depending on the cell type, developmental stage, or by any other environmental conditions. And it's also important to know that when the genome is defected, it's the proteins that are ultimately get affected, since they are the actual functional molecules in the cell. And even from the one defected genome, many proteins can be affected. So that's why proteomic studies are quite important and informative to understand how things are changing in the cell and how things are working. So we can say proteomics is a large-scale study of proteins, and it has many different branches, such as functional proteomics, structural proteomics, protein-protein interactions, and it has been used in various types of study areas, including cancer proteomics, drug discovery, and disease mechanism, for instance. But the main goal of the proteomics is to identify all the proteins in the cell and identify the changes in the proteome. And it has mainly 3 steps, which are sample preparation and data acquisition, where you use mass spectrometry, and then data analysis, where you do use bioinformatic programs to understand what does your data mean. And these steps are all connected to each other, and they are all affecting each other. And according to the study, according to the final aim, you can modify these steps. So this is the classic workflow for sample preparation. You start with cells or tissues. You extract the proteins out, you digest them, you fractionate them, and you run them in mass spec. But there are some issues with the classic workflows. They're not able to handle the sample complexity well because there are a wide range of proteins in the cell, and their abundances are quite different. And you can see from this graph coming from a serum sample, there are proteins that are highly abundant, and there are also proteins that are low in abundance. And the proteins which are highly abundant will be identified and also quantified more easily than these proteins, because the sensitivity for detection will be affected by the purity in the sense of the proteins which are highly abundant. Okay, there are some solutions for this problem. People are using different techniques. For example, there are things you can do at sample preparation step. For instance, people are using different columns, such as reverse phase cation exchange, and sometimes they use it in a tandem manner. And you can go for an enrichment. If you have a protein of interest, you can pull the proteins down. You can even change the way you do the lysate. If you're mostly interested in membrane proteins, you can just go for them. There are also things that can be done in mass spectrometry analysis. For example, you do longer runs, you will get more identification. It can be 1 hour, 2 hours, 3 hours, depending on your project. And there are also things that can be done in data analysis. So I mentioned the protocol I developed. So besides getting an increased identification, I also wanted my protocol to work well in comparative studies. But the problem with the comparative studies is, okay, you can check this scheme. So this is showing the proteins in the sample, and this is the proteins that can be identified with the current techniques. And this is the proteins that can be quantified. And the factors that were important in the identification are also applies here. Like protein abundance and complexity, but techniques for quantification and their accuracy also matters. So for comparative proteins, there are 2 main techniques. One is label-free quantification, and the other is labeled quantification. So in label-free quantification, both of them you start with cells or tissues. You extract the proteins out and you get the peptides. But in label-free quantification, you do 2 different runs. You start from 2 different samples, and then you get the data analysis. And what you'll do after is the comparing the spectra you have. While in labeled quantification, you do only 1 run. Your control and experimental are in the same sample, and you do only 1 run and you get 1 chromatography. So although this kind of quantification is making the data much more complex, like doubling the complexity, they're considered more accurate, so we wanted to continue with the labeled quantification method. And in labeled techniques, there are 2 main well-used and well-known techniques called ICAT and SILAC. In ICAT, you use chemical labeling reagents to modify the cysteines, and then you do many steps after you combine the sample. And then there is this denaturation step, and then purification steps. It's taking time. And while in SILAC, it's the stable isotope labeling kind of tagging. And in this kind of technique, the isotopes, they're already in the medium, and they will be incorporated in the peptides metabolically. So they're in there, they're stable, and the mixing of the 2 samples will be happening right after you lyse the cells, and then you do the mass spec analysis. So there are advantages and disadvantages of these 2 techniques. So in ICAT, since it's only labeling 20% of the peptides, it will reduce the sample complexity quite a lot, like 80%. And this will get you an increased number of identification. But the amount of starting material in ICAT is important. And there are 6 steps before mixing. So that's affecting the accuracy of your data. And there are affinity binding chromatography and purification steps. It's taking time. And most importantly, the quantification is coming from usually 1 peptide. While in SILAC, there are no chemical labels, and the labels are stable. They're already in peptide. And they're mixing the samples very early, so it's accurate. Although it's labeling all the peptides. So it will give you a good quantification from most of the peptides. But since it's increasing the complexity, it will affect the identification. So my aim was to combine the advantages in 1 technique. So it goes like this. I start with cells, I'm labeling them, and I'm mixing them. This part is like SILAC. Then I do cystic acid oxidation. This is like in ICAT. I'm doing some cystic acid modification, but it's just taking like 1 hour. Here I'm taking the advantage of cysteines that can be turned into cystic acids here. And this will create a supernegative part in the peptide, which can be selected out by a mixed mode exchange column. And then there will be mass spectrometry analysis and data analysis. So this is how it looks like actually. It's an offline thing. I'm basically doing it in a pipette tip. So I'm using a reverse phase resin here just to hold everything up. And then I'm pushing the oxidized and digested sample in. And I'm adding a couple of steps to make sure that they're binding and there are no unspecific binding. And then I'm eluting the peptides with cystic acid. So what are the benefits? Since I'm labeling all of the peptides, like in SILAC, I will get a good quantification for most of the proteins, and it will be an accurate quantification. And I'm continuing with the chemical steps of cysteine oxidation, and it will enable me to enrich the peptides with cystic acid, which will reduce the sample complexity after to get an increased identification. So we first tried it with a strong cation exchange, and it failed actually because we were expected to see peptides with cystic acid, which are in a red column, will be here, and but most of the things were coming in the earlier steps. And what also important, we were also not seeing the many cystic acid containing peptides, and we had a problem with quantitatively modifying cysteines. So I had to figure out what kind of chemical I can use to oxidize them in a short time. So we used the different oxidants, and they also should be compatible with the MS. And we had to decide whether it should be in solution or in gel. But the most important thing was how to enrich the cystic acid peptides, what kind of chromatography should be, and what are the conditions for the chromatography. So for that purpose, I chose a specific chromatography called ERLIC. It's an electrostatic repulsion hydrophilic interaction chromatography. So in this kind of chromatography, the strongly acidic peptides will be retained more strongly. In my case, they are the peptides with cysteic acid, and the basic peptides will elute earlier. But still, I had to alter the conditions for this chromatography. I didn't use the classic ones. So you can understand better here with this scheme. This normal reverse phase, and peptides with cysteic acid and also peptides with non-containing cysteine will be attached. All the acidic peptides will be attached in here. While in this kind of chromatography, I'm expecting peptides with cysteic acid will attach, and the rest of them will be repelled. So then we moved to weak anion exchange, and the results got better. So we were seeing the enrichment. This is the total number of the peptides. This is the peptide with the cysteic acid. I just differentiate them by singly cysteines and doubly cysteines. So this is the total amount of peptides with cysteic acid, and we were seeing the enrichment. However, there were a little too many steps, and we wanted to know if we can shrink the steps down, and if we can also save the peptides with cysteic acid that will already out in the flow through and wash in the next wash. So we had to play with the many factors, and one of the most important was the pH. I used different pH starting from 2.5 to 8, and for example, this is for the pH. The pH was 3.5. I mean, there is an enrichment, but however, it's not very efficient. However, when we play with the pH, we realize that at pH 7, we are getting a good enrichment. This is the total amount of peptides. It's the peptides with cysteic acid. And you can see in elution, most of them are peptides with cysteic acid. Another factor that is important is overloading. So this graph, okay, this is the experiment that I use 1 XPSA. But what happens if you cannot arrange the amount of starting material? What if you use something more? Well, this is happening, sorry. So when we increase the amount of BSA, like doubly, there is still an enrichment, but you will start, since you push the column too much, you will start losing peptides with cysteic acid in earlier steps. In order to fix this, we decided to increase the volume of the resin. For example, here I used only 1x resin, and the conditions are same, the amount of starting material is same, but when you increase the volume of the column, it's affecting the enrichment, because once the column it reaches its capacity to hold the peptides in, then whatever you do after, you will be losing the other peptides that should stick. Okay, another good result was the minor overlap between the experimental step. Okay, this is showing the peptide list in the flow through, in wash, in the second wash, and elution. So the peptides that I colored, there are the peptides unique to the experimental step. For example, these 5 only seen in flow through, this 2 only seen in wash 2, and etc. And the uncolored ones are the common ones. For example, this also seen in here, and these are also seen in here. So this shows a really good fractionation between the experimental steps, which helped us in a great sense to get an increased identification. So you can see also in the graph, this same data. We also tried this in BSA and also in H1299 cells. And this is the total number of the peptides that are identified in 3 steps. And the red columns are showing the common ones. So they're really minor, so it's helping a lot also with fractionation. So this is the last version of the technique. As you can see, it's very short, and we only have 3 fractions, so we have enough time to analyze all of the samples, so we're not actually losing any peptides or any proteins in the previous steps. So I start with sample loading on the gel, and I'm doing the in-gel digestion, and then I'm doing the performic acid treatment 1 hour, and I'm going the mixed mode chromatography that I do on the pipette tip, and then we go for MS analysis. So as a next thing, we wanted to validate the protocol using a biological system. So besides proteomics, I was also interested in neurodegeneration and cell senescence. And there are many different models to study these disorders and conditions. For example, one is expressing the disease protein in cell or animal models, genetic models. However, they're takes time to develop. They're very complex. It's hard to get them. And it's difficult to reproduce the same model. So that's why I want to use a chemically induced in cell culture, because they're easy to develop, they're simple, and they're easy to reproduce. But for the most important thing, we chose them because they're proper for validating the method. So as a chemicals, I choose two different pharmacological compounds called serotinol and resveratrol. These compounds have been proposed to use for both cancer therapies and the therapies for the neurodegenerative disorders. They have an effect on sirtuins, especially class 1 sirtuins, which have a robust deacetylase activity. And SIRT1 is known to prolong lifespan in yeast, and it's included in neurodegeneration. And so that's why we wanted to see the effect of these drugs on cells and with the method. And serotinol is known to induce senescence in cancer cell lines, so we want to try with H1299 cells first. So what I did, I get the H1299 cells and I plated them in light medium and heavy medium containing the heavy larginine and icing. And I waited at least 5 divisions, which is like 1 week. And then I did the serotinol treatment. So this is how it was like. I did this is the control, and I used 200 micromolar serotinol and 300 micromolar serotinol. I only treated the cell for 24 hours. Then I waited 10 days, and then I did the acylic beta-gal staining to see if they're positive for it, and it can show the cell senescence. So in fact, there was a change in size and shape, and they became beta-gal positive in a great sense. So we checked the positivity for beta-gal, and with the 300 micromolar treated sample, the positivity was about 90%. We also wanted to check the division of the cells if stopped or not, and you can see the control keep growing, and 200 micromolar treated cells, it's stopped until the day 4, but then it's somehow surviving and going. And but in 300 micromolar treated one, the cell division was arrested for 10 days at least. So after that, I lyse the cells, treated with the serotinol, and do one-on-one mixing, and then do the in-gel digestion, the hybrid method, and mass spectrometry analysis. So I use many different mass spectrometry analysis engines, such as Mascot and Extendum. These are very famous. The way they work is a bit different, since they're using different algorithms to analyze. So the results are a bit different. But since Mascot is more strict, we wanted to stick with it. But still, we use the tandem results as well. So first thing I wanted to see, since the serotinol is affecting the acetylation pathway, I want to see if the total acetylation number will change or not. And in fact, in the serotinol treated sample, the acetylation went up high, like more than 40%. And this showed how the serotinol working in the cell. It's a proof that serotinol worked in the cell. And it's also further proof that the method is working. So what is also very good, that the total identification number increased a lot. With the classical method, we were able to identify 1,000 proteins. In my method, it went up to 5,000 proteins, and also the peptide identification increased a lot. And what's also good is that we found out this method can be also used for quantitatively mapping the oxidation and phosphorylation sites, and like comparing the classical method and my method, the oxidation identification of oxidation site increased 70%, and in phosphorylation we have an increase 100% for identification the phosphorylation site. Then after, I moved another part in the project to validate and quantify the proteins. And for that, I used the program called ProteiIQ. This is used for quantify the proteins, whether they're labeled or label-free. And this is good for comparing the sets you have, the groups you have, and it's also giving biological annotation of the proteins that you have. It's also worked for statistical validation using the false discovery rate calculations that you do with the decoy research. And you can further analyze your results in a different manner. For example, it can be peak dependent, giving info about the peaks. You can compare the peptides, what happened to peptides in the level, or if they're having any PTMs, etc. And you can compare the protein levels, and you can compare the groups. So since I started with SciLac, the quantification was going to depend on the label quantification. And so in this kind of quantification, you get a relative protein expression. For example, this graphic showing the two peaks belonging to the same peptide, tagged with light and heavy. So basically, it's the changes in the amount is measured by the different intensity of the peaks belonging to the same peptide, and it's just making some calculations to understand what is the change. So this graphic is calculated by that. It's with ProteIQ, and in this graphic we see that we quantified nearly 4,500 proteins, and out of those we identified 140 protein groups that are upregulated, and 37 proteins are downregulated, and we see that most of them are not changed, and this is what you want to see and expect in mass spectrometry result. To further validation, I put my data on David database to get the functional annotation chart, and as here also we were seeing the acetylation pathway is coming first, and this was the further proof that the method is working. And there were also other pathways that were important. This is a list of the proteins that downregulated in the search-null treated experiment. So when they like this, they don't mean much, but what you can do is get the protein-protein interaction pathways using programs called, for example, like String. And in this kind of pathway, it will help you find an interesting relationship between proteins. For example, here, these proteins are mostly have a relationship with each other, so this will be further studied. So the second cell system was the neurons. So for that, I used the normal tissue dissociation cell culture. I first started with P-mod mouse. It was easy to dissect. It was easy to identify where is cortex, where is hippocampus, and so on. But that experiment mostly failed. The cells were not able to stay viable, and they were dying after. Then I moved to embryonic stages. I used many different embryonic stages, starting from 10.5 to 17.5. And for me, 13.5 was the best model that I can go with, because in this stage there are less glial cells, so you have less contaminant cells, let's say. And it's better for primary culture because they are staying viable for longer times. So I started with seta mating, and then I'm harvesting embryos on E13.5, and I do the cortex dissection, and then the tissue and tissue dissociation and plating the cells. And then I do the RSU treatment on the second day to make sure that I will inhibit the glial cell growth and fibroblast growth. And then I did the search-null resveratrol treatment after the viability assays. So there are some issues using the neurons in mass spectrometry. For the first thing, obtaining a homogeneous neuron culture is important, because when you treat it yourself, you will never know the effect is coming from the fibroblasts or the glial cells. So you have to make sure that they are inhibited. So for that, I use RSE, and it was working well for me. Another problem is reproducing this exact same culture. So even I am successful at choosing the right day and finding the E13.5, there are no noticeable differences between the noticeable differences in embryonic development from litter to litter in the mother. So plus, what we also wanted to do is using the label-free quantification for neuron culture, because the thing is, SciLac is preferentially labeling the proteins that having the high turnover rate. And in neurons, they're not dividing. So, and for example, in H1299 cell, the unlabeled proteins will be diluted out from the sample because there are many cell divisions. However, in neurons, it won't happen like that. So it can be misleading data. So we decided to use the label-free quantification with my method. So in label-free quantification, of course, there are no labels, no tags, no isotopes. You're not doing anything. So you start with two different populations. You lyse them, you fractionate them. You do two different mass spectrometry analyses, and you can compare MS or MS results after. So there are different calculation methods. One is like area under core or spectral counting. So in my experiments, we chose the spectral counting method. And it's built on the idea that the number of spectra identified for a protein is directly correlated with the amount of the protein. So that's why the starting amount of the two experiments, it's very crucial. You have to really start with the same amount, and you have to treat your sample all the way same to make sure that your quantification will be accurate as much as possible. So for that, I plated cells, and I waited at least 10 days in the plate so that neurons had the good growth. And then I did the sertinol treatment. And I like the cells. This time there's no mixing, and you do the in-gel digestion and the hybrid method and mass spectrometry analysis. So this is the very preliminary result of a small data set. This is just coming from one band, and you can see the proteins identified in the groups, different proteins and the common proteins, and I also get the protein relative expression change, and you see still not much of a thing changed as it should be expected even in a small data. So this is also ongoing experiment. So conclusions. So this method is quite useful to getting an increased number of identification, and this is accomplished by the sample fractionation and enrichment steps that I made. It's accurate and straightforward protocol for comparative studies, and it's effective for giving reproducible data. So after sertinol treatment, this total acetylation number is increased as expected, and we see it in the results. And with the hybrid method, approximately 5,000 proteins have identified, and 90% of them are quantified. And in ProteIQ result, there are 140 protein groups that are upregulated, and 40 proteins are downregulated. And it does not require SILAC for the protocol to work. So since SILAC is not required, this method can be used in any kind of cells. And this technique improves the identification of the post-translationally modified peptides, especially those that increase the acidity of the peptides like phosphorylation, oxidation, and acetylation. And it can be quite useful for the binary comparison, as it has been validated by the results. So the future work will be finishing the mass spectrometry runs, and I will have to have the repeats for in mouse models and also in H1-99 cells. Then I'll be validating the changes by Western blot. Then we'll go for checking the biological relevance with the cRNA or overexpression experiments. So I'd like to take my lab and mic, and thank you for your attention.